Bob Hopkin. I am a clinical geneticist at Cincinnati Children's Hospital, and I'm honored to have been asked to speak to you today about emerging therapies for Fabry disease. We're specifically going to be focusing on gene therapy. I am speaking on behalf of the Fabry International Network, and these are my disclosures. Now, currently we have a number of therapies for Fabry disease, but they all have downsides. One is that you have to take the medicines over and over again. So for ERT, you have IV infusions every couple of weeks. You have the risk for infusion reactions. And there are some aspects of Fabry disease that aren't very well targeted. For example, it doesn't completely stop progression of heart or kidney disease. And the chaperone therapy is only suitable for certain patients. Depending on the mutation, it has limited ability to control the pain, and probably also does not completely arrest the heart or kidney disease, although both of these medications do clearly benefit patients in a significant way. Other therapies such as kidney transplant or heart transplant have high risk for serious complications and require lifelong sustaining immune suppression and other things. Plus, they only correct the Fabry disease in the organ that's transplanted, not throughout the person's body. And finally, some of the substrate inhibition, which is an experimental therapy, looks like it has some promise, but it also has some clear limitations. For example, there's a tendency for drug-drug interactions, and it likely will, as with the other therapies, require lifelong therapy and have some continued disease progression even with successful treatment. Because of that, we're still in the market for newer treatment. Now, we have a lot of experience with enzyme replacement therapy, and we know that currently we're treating people usually at an average age in their 30s. At that time, about 50% will have significant kidney disease. Even more than that, about 80% will have cardiac involvement. There will be white matter lesions in the brain, GI problems, etc. We want to treat people earlier, but we also don't want to disrupt people's lives unnecessarily. So what do? Well, we need a therapy that we can use that's going to be less of an impact, less of a burden on the patient's life. Now, Fabry disease, with the current treatments, most of the patients report some pain. Many of the patients report recurrent diarrhea, constipation, bloating, nausea, or just abdominal pain. And the current therapies haven't really addressed those needs as well as we would like. Again, we're still in the market for a better treatment for this disease. All of life uses the same instructions. The genetic code is the same for bacteria and viruses and plants and animals and every form of life that we know of, which means we can learn about that code, which is DNA, learn to manipulate it, and then potentially develop therapies using DNA. Now, it's not as simple as just cut and paste from one species to another or from one person to another, because humans, chimpanzees, and monkeys have more than 90% of their DNA in common as far as what proteins are coded for in the genes. But it's easy to tell the difference between a person and a monkey or a chimpanzee. And the difference there is in the pattern of expression and the regulation of the expression of the genes. And that is an important part of the way that our system works. So the principles of gene therapy is you find the gene, you isolate the gene, you make multiple copies of it, and then you have to deliver that gene into the cells of the person that you want to treat. There are two different categories of gene therapy. One is germline, and that one, it targets all of the cells in the body, including the reproductive cells, so that the change that you put in could be passed to next generations. That is thought to be more dangerous and is therefore currently not being actively pursued in most settings for use in people, although there are hopes that we will eventually get there. The other category is somatic cell therapy, where we correct the cells in the target organs in the body and have them be able to produce the gene product that's missing. In the case of Fabry disease, that's alpha-galactosidase A enzyme. And that cannot be passed down from one generation to the next. So if a new person is born with that disease, they would need to be treated, and each generation would subsequently still require treatment. But if it's gene therapy, hopefully the treatment would be simple, well-tolerated, and very infrequent. The requirements for gene therapy is you have to be able to package the gene, and that has to include the promoter to the gene that turns it on and off, the coding sequence that tells you what amino acids go in the gene product, and then the post-translational instructions that fold, shape, and modify the proteins that are coded for by the genes so that they can do the functions that they are supposed to efficiently. In addition to having that information encoded in the DNA, we have to have a delivery system that will take the gene into the cells and deliver it so that it can be used by the cells. And we do that either with viruses, RNA viruses or DNA viruses can do that, or we do it with non-viruses, things like liposomes, or just putting in naked copies of DNA. So if we do that, if we get the gene with the instructions and a vector and we can deliver it, ideally what we're looking for is a direct molecule that gets into the cell, is given by a single administration, and then functions throughout the lifetime of the person. And that ideally does not have any potential to cause side effects or pathogenic impact. And if possible, we should have the ability to turn up, down, or even off the gene product. Now, Fabry disease has been looked at as a possible candidate. There are some challenges to treating Fabry disease with gene therapy. One is that you would have to get the enzyme produced in the body and delivered to multiple different tissue types and organs. It's a multi-organ disease. You need to get the treatment to the kidneys, the heart, the brain, the peripheral nervous system, the eyes, the ears, the intestines, etc. This disease progresses slowly, which makes measurement of your success with gene therapy more challenging. So benefits that Fabry disease has is that the enzyme is not bound. The enzyme is relatively small, which makes it easier to package into a gene therapy, and that overexpression of the enzyme does not appear to be toxic. The disease is rare, but it's not so rare that we don't have enough patients that we can do good studies. Thus, overall, it looks like Fabry disease is a good chance. Now, when we choose vectors for delivering gene therapy, we want them to go into the right tissues. And that is the topic of tropism. So different viruses attack different parts of the body. And you can select viruses that will, or vectors that will target neurologic tissues, immune system tissues, etc. In the case of Fabry disease, you need kidney, heart, brain, and neurologic targeting in order to be successful. And that means you go to this library of viruses, and each of those types of viruses has multiple individual viruses, and they will all have varying ability to target the specific tissues. So you pick the one that's most likely to succeed, and then you package that. But you don't just put the whole virus into a person. That would cause severe infections and could, in fact, even be life-threatening. So what we do is take the virus, cut out the genes that make the virus cause infections and inflammation and make you sick, and drop in place of those genes the gene of interest, in this case, the GLA gene that makes alpha-galactosidase A. So that's the project, that's the goal. And now we're going to talk a little bit about some specific attempts that are being worked on in people. Now, for the most part, receiving gene therapy is a pretty straightforward process. You get an IV, we run the vector suspended in fluid into your body. It's very much like the experience of getting an enzyme infusion, except that you only do it once. Now, there are multiple groups trying to make gene therapy right now, but we're going to just talk about a few of them. The one that's been used the most at this point is this ST920. It is an AAV virus-based delivery system that is liver tropic. So basically, the attempt here is to put the GLA gene into cells in the liver, cause the liver to produce a large amount of the enzyme, which then gets pumped out of the liver into the bloodstream and circulated to the rest of the body. This has been done in around 10 people. It has been well tolerated with no serious side effects and very few mild side effects to date. There has been a high level of enzyme production in the patients that have received this therapy, but it's still too early to assess how well it's impacting the progression of the Fabry disease. The initial preliminary findings are that it is helping the patients, but we still have a lot of work to do to get to that point. Okay. Another approach that's very similar. So the first one, you just kind of take over the liver and make it into an enzyme factory. But the liver isn't really damaged very much or involved very much with Fabry disease. So some people thought, well, what if we could make not only the liver, but the target organs make the enzyme? would that lead to better delivery and more efficient treatment? So they selected AAV9, which does go into the liver, but it also gets excellent penetration into the kidney, heart, and brain. This one, like the other one, requires overexpression and requires the enzyme to get into the circulatory system to be then shared with the other parts of the body. In the animal models, it showed wide distribution of uptake of the vector and expression of the genes that were targeted in the animal models. So this one is looking promising and is getting ready to be used in people. And it also looks quite promising, but it is too early to really make a decision about, is this the best way to go? This one is quite different. So the first two, we just took the GLA gene, dropped it into a vector, delivered it into the cells, and then the gene turned on. For this one, they said, wow, this particular mutation that's listed on the slide is quite common, particularly in Taiwan. And we want a treatment that will treat these patients in a simple way and with minimal fuss. And so they said, maybe if we not take the GLA gene, but if we put in some instructions for how to edit the gene that's already in the body, maybe we can convert the cells so that they will make their own enzyme. And they have tried this in mice and put in instructions to take that mutation that's listed on the slide and edit it so that it's no longer abnormal, that we correct it and make it like the enzyme our body is supposed to make, and then see if it produces. And in the mouse, they were able to get adequate enzyme production and normalization of the Fabry-related biomarkers. So that one is conceptually cool, but not necessarily highly practical. The next one I want to talk about is a variation on that same theme. People were thinking about it and said, well, if we just drop in the enzyme the way it's normally made in the body, that might not be the ideal because the enzyme is inactivated in the blood. So we lose a lot of enzyme. So they said, well, why don't we modify the GLA so that it produces a protein that does what alpha-galactosidase A is supposed to do, but that it stays stable as it transitions through the bloodstream into the cells. And that one is also being done. So they have an augmented gene product that is for the purposes we're using it for better than what our bodies would naturally make. That's a very exciting concept, although it may have some risks associated with it, but it looks like it could be an excellent way of delivering medications to targeted tissues in the body in a more efficient way. And finally, we have the lentiviral approach. So the first ones have all been delivering the enzyme or the vector into the cells in the body. For the lentiviral, you take stem cells out of the body into the laboratory, treat them with the gene therapy, test the cells to make sure they're still healthy, and then infuse those cells back into the body. Now that one has some benefits in that you can, as the cells divide, more and more of the cells in your body would be able to produce enzyme, but it also requires some more aggressive immunosuppression to get the stem cells to make room for the stem cells in the body so that they can reproduce and spread throughout the body and produce the enzyme that we want. So this one has also been used in people. It showed some initial promising results, but when they tried to expand the protocol so that they weren't just doing one batch at a time for one person to be treated at a time, they had trouble maintaining high levels of expression. So this one is currently on hold, but it is still an important approach because the treatment actually is done outside of the body, which means there should be less risk for complications of the gene therapy. There's some other things that are gene therapy like that are also worth keeping an eye on. One of them is RNA-based therapy. Now that one would have to be given over and over again, but RNA, especially if delivered by liposomes, it has a low risk for infusion reactions and is therefore likely to be as efficient or more efficient than enzyme infusions and less likely to result in infusion reactions, and may get into some tissues that are not well targeted with ERT. So that's worth at least keeping an eye on. With the pandemic, this one has been kind of on a back burner for the last couple of years. So in conclusion, there's a lot of work being done to develop gene therapy, just in general, and specifically for Fabry disease. This is likely to become a true treatment option sometime in the next 5 to 10 years. We do not know how the utility or the impact of that will compare to the current treatment options, so we'll need to keep an eye on that. We do know that there are some risks associated with gene therapy. Those are being addressed, and one of the reasons that it's going to take probably several years to get this to market is to make sure that the treatments are in fact safe and that they will have long-term duration. Hopefully, one treatment could last a lifetime, and we will need to make sure that we get adequate tissue and cellular penetration. So this is, I think, an exciting area. I have not mentioned any specific groups or companies that are working on gene therapy. If you wish to get that kind of detailed information, I suggest you go to the people who are providing the care for your Fabry disease and ask them. If you prefer, you are welcome to try to contact me through email, and we will try to get you some information as needed. But I think you would be better served to have those conversations with the people who know you, know the stage of your Fabry disease, and know what's available in your area. Thank you. We will end here, and hopefully we'll have some good discussion in the question and answer period.